The report that I'm going to be talking about in the next few minutes has to do with, as the first screen says, transgenic rootstocks mediated protection against transformed scions. So what we're really doing is we're now beginning to test the potential for cross-graft protection of Pierce's disease susceptible, in this case, a Chardonnay scion, against the development of the symptoms of PD by the expression of dual combinations of 5 previously identified PD suppressive transgenes. And we're doing that into 2 adapted rootstocks, which we'll talk about. And so we're really assessing both the potential cross-graft protection of the non-transformed scion and the effect of the transgenes to possibly protect the rootstocks against downward movement of the bacteria in the perennial tissues and subsequently plant death. And these are all being compared to equivalent combinations of untransformed rootstock scion combinations. First, a brief history of how we got to this point. The quest for this probably began around the time that Craig said that this disease became a real issue, and that was around the year 2000, when a number of researchers listed here, Steve Lindo, Abhay Dandekar, the late John Labovich, and myself, looked for genes that had the capability of perhaps suppressing at least PD activity. You can see on the left side of this screen, there's Pierce's disease symptoms in the first field trial that we did, which was under USDA APHIS control. And this field trial went on from 2010 to 2016. What it did was it confirmed the presence of disease suppressive transgenes when they were expressed as single genes as whole plant transgenics. And on the right, there's an example of a Thompson seedless plant that was injected with the bacterium Xylella fastidiosa by inoculation. And the one in the foreground is clearly dead. And the one in the background where the red arrow points to was equivalently inoculated, but in fact persisted without any symptoms. So this was the beginning then of the kind of things that we're now going to be talking about. This was discussed with the Pierce's Disease Control Board. They were favorable with respect to proof of concept in the first trial, but then agreed that this trial could be terminated, and then we proposed a second project, which was going to be field testing these transgenic rootstocks. Following the APHIS's regulations, the field was burned and all the material was destroyed completely. And so then we moved to the next step. So the second experiment, as illustrated here, was to test whether we could take combinations, pairs of the genes that had been previously looked at in the field trial, express these in adapted rootstocks, and whether or not this resistance that they were able to produce in the whole plants could in fact be transferred across a graft union to the untransformed PD susceptible scion, in this case Chardonnay. And the 2 rootstocks that we chose were 1103 and 10114. This identifies the first critical step, and that was to develop an expression binary vector that could insert simultaneously into a plant cell, which could then be regenerated into a whole plant. This is not a trivial exercise. In fact, it's unique with respect to the ability to move 2 genes simultaneously into these adapted rootstocks. Now, Jim Lincoln was the one who did this, and without him, it probably wouldn't have occurred. So the vector was created, and it was successful with respect to the ability to transfer dual combinations of the 5 genes that are listed here. And they have different presumed biological activities, so we're really complementing activities then with respect to their expression in the rootstocks. We also spent a lot of time assaying all of the transformed cells that were provided to us by the transformation facility at UC Davis, and something like 67% of them had actually both of the genes present in the tissue and were being expressed. And there's an illustration on the right what these little baby plants look like when we received them from the transformation facility. These plants were then grown in the greenhouse, as you can see in the top portion of this slide, in order to provide cuttings. And so each of these plants was then capable of producing a number of cuttings for clonal propagation, and then subsequently they were bud grafted with the wild type Pierce's disease susceptible Chardonnay, and with these 2 genes at a time. And you can see the little bud grafted plants in the foreground, and then some of their progeny in the other side, part of that slide. Again, just an example of the greenhouse then cultivation and growth of Chardonnay scions grafted to these rootstocks in preparation for the field planting, which is again going to be done under a U.S. APHIS approved and highly regulated site. The permit for this is held by Dr. Abhaya Danikar, and they're responsible for making sure that we provide APHIS with the information they need to make sure we're all the rules, and there are many. In 2019, we finished the planting on the left of these two panels, the transformed rootstocks with the untransformed Chardonnay, and also with the control plants, which were untransformed, the same genotype, same rootstocks, same scions, so that acceptable control against anything that would be viewed as suppressive versus disease symptoms. The panel on the right illustrates these cordon trained plants in May of 2020. They are in fact trained in this way by commercial standards so that we could, if necessary, if it turns out that anything works, be able to actually measure fruit production and other kinds of economic characteristics on these plants. That was all completed in 2019. You don't expect to read this, but this is just simply for your information if you want to look at it any closer. This is a list of the 721 plants that are now in the field, growing, and are, as you'll see on the next slide, they in fact are at the point now of being inoculated. So the infection then of these plants is in fact carried out by simply inoculating each of the plants, number of shoots on the plants, with something like 20,000 shoots, 20,000 colony forming units or bacterial cells per inoculation site. This is done by stem puncture inoculation. As you can see in that little tiny picture in the center, a little droplet is placed on there, and a needle is punctured through that or from the opposite side, and the vascular system, of course, will suck that droplet in with the bacterial cells along with it. Now, we anticipate inoculating a number of about at least 5 sites over the time to make sure that the is established. In fact, we've already previously primarily done that, but I'll say that in a moment. The other thing I need to point out is that there's been the plant spread of the bacterium in the previous field tests. Once the bacteria was implanted into a specific plant, it stayed there. It moved in the plant, and in some cases it killed the plant, or in other cases where the disease suppressive transgenes were in fact functioning successfully and the symptoms of PD was suppressed. But there's been no plant-to-plant movement of the bacteria, which means that this experimental project, it has a very tight experimental control. We don't have any spurious movement of the bacteria at the plant. The bacteria is only present where we put it, and it seems to stay there. The image on the left is an example of the 721 plants that are out in the field right now, and it shows prior to inoculation of the individual canes, which were carried out in July of this year. So the plants that are to be inoculated have been inoculated. The photo on the right shows sites of inoculation. This was an inoculation that was completed on July 23rd of this year, and this picture was taken in November after the leaves had begun to drop. On the next slide, the data collection and analysis is summarized here. There's a lot of text here, and I won't go through all of it, but what we will do and are doing and have done actually is to confirm that the infection was complete in samples that were taken in October of 2021 following the July inoculation. These tissue samples are then taken to the laboratory under tightly controlled conditions, as it turns out, again, APHIS controlled. And then the material is extracted, and by qPCR procedures and a rather laboriously determined, but now relatively very effective means by which we can recover the bacteria from these tissues and actually quantify and qualify them. So we've confirmed that the inoculations that were done in July are in fact effective. The bacteria is in the tissue, and so now samples will be taken subsequently. And as the plants begin to push again in the spring, we will begin to look for symptoms associated with the control plants and the ones that are in fact the treatment plants. As it also says, the very last point here was that we'll manage these to commercial standards. So if it turns out that measuring fruit yield per plant as the disease progresses is something important, we will be able to do that. We're set up to do that. This summarizes the steps that have been taken and the timeframes in which they were both proposed to be done and in fact were completed in a timely manner. We've been able to follow this time in spite of the constraints that have been placed more recently by COVID on our ability to actually have people in the lab, and it's been tightly restricted, but we've been able to manage to work around that. And we've been able to follow this timeline to the point where we are still on time. You can see that we began the transformations in 2015, about the time that we were winding up the original trial. These plants were then taken to the greenhouse. They were analyzed, as I said previously. And then the grafting took place, and ultimately we also did some test inoculations in the greenhouse to make sure that the bacteria that we were using were in fact infectious, and that we could in fact extract from the tissue and measure the amount of bacteria that as they were present in the plant and as they may move in the plant. So we are now at the point of this with 1 minute and 3 seconds to go. I think this will conclude what I have to say with respect to this potential cross-graft protection from these transformed rootstocks. And I think I can leave it at that. We are still on target, and we expect to continue this program as it was outlined in the beginning and as we are now seeing it take place. So thank you very much.
We do have a question for you, David. Is any work being carried out to facilitate the acceptance of transgenic plants for wine production?
It's an excellent question, obviously. Clearly, in the first trial that we did, the whole plant transgenics, that was never going to fly with respect to any kind of production and sale. That's why we moved to the rootstocks. And what we hope is that if, in fact, the rootstocks are the end product and the end where any transformed material, transgenic genes are in fact present, that and nothing other than protection moves to the untransformed scions, that this is could be something that would be more readily accepted by the industry. And that's the whole goal behind this. This is what the Infectious Disease Control Board was interested in, was to see if there was any way that we could instill protection with these transgenes and yet not in any way have the scion, which is the fruit producing portion of the plant, in any way transgenic. So I'm not sure that answers the question, but that is in fact the basis for why we're doing what we're doing right now. And we'll see how it goes. First of all, it has to accomplish something and with respect to protection. And then these plants this process then can be in fact regulated, and I can see how it goes. So it's a great question. It's the central question with respect to any kind of transgenic changes in plant tissue, but we don't know the answer to it at this point. But we certainly have that as a foremost in our minds with respect to the future utilization of these kind of materials.